Hello and welcome to the Huxley Speaker Series. My name is Stefan Freeland and I am the host today. And as usual, we'll be doing questions at the end. You can submit questions via Q&A or the Zoom chat. There'll also be a Zoom poll coming at you, so get ready for that as part of the presentation. It is my pleasure today to introduce our guest, Dr. Drew Harvell. Dr. Harvell is a professor of ecology and evolutionary biology at Cornell University. Despite her geographic location most of the year in upstate New York, she also does research on the Salish Sea and has a more local connection as well. So today we'll be hearing about her this coast studies as opposed to her work in upstate New York. She also has a new book out, which we'll hear a little bit about, called Ocean Outbreak. With that, it is my pleasure to turn it over to Dr. Drew Harvell. Welcome.
Great. Thank you so much, Stefan, for inviting me and for this amazing series. I was sort of looking through some of the other talks, and I'm definitely going to be listening in to some of the others. In terms of my interests and my research, actually, what you can see behind me is False Bay on San Juan Island. That's where we live. And I'm looking across to the Olympics there and kind of up Georgia Strait to Canada. So the Salish Sea is sort of the heart and soul of where we live and one of my big priorities. So this really means a lot to me to be here today. So the talk I want to I see there's quite a few of you online. I want to congratulate you. This has been an absolutely crazy time, a crazy week. There's too many things going on to pay attention to. You all deserve incredible credit for sitting down and being here today. So I'm delighted to be with you. I'm going to talk about ocean outbreaks, and I call this of foundation and keystone species because I'm an ecologist, and my greatest concern is how infectious disease is affecting some of our really important constituents in our marine ecosystems. Not only am I professor at Cornell University, but I'm also a resident researcher at Friday Harbor Labs. And so that's really kind of explains the interest in Salish Sea. We've been working on ocean disease for, well, several decades, but the idea to do this book really grew out of a National Science Foundation project that I led called the Ecology of Infectious Marine Diseases, and it's a research coordination network. And this was a team of over 60 scientists that met regularly and worked on all kinds of issues of infections and how they spread and propagate in the ocean. And of all the work, I think that came out the most clearly from this project, it was the effects of warming climate and disease. And indeed, all that I'll talk about today will be in that same realm. So I just have included here, since this is going to be recorded, a few of the papers that other team members in our research coordination network produced over the period of our grant. And it's really this collaborative work that led to my writing that book. So it feels like all of us wrote it. And I'll come back to that towards the end. So the focus of my lab is the ecology of host pathogen interactions. And in addition to being important in their own right, we also think of them as sentinels of change in ecological interactions. And I hope as we go along today, you'll come along with me to see that. So this Venn diagram here has 3 bubbles, and I bet you can guess how I'm going to populate them. In all of our interactions, there's a host. There's an infectious agent, and there's an environment because we're ecologists. And these interact to produce what could transpire as a disease epidemic. And so today I will briefly recount some of the work we've done with corals, but mostly talk about the more local projects with eelgrass and seastars. And so my talk has kind of 3 parts. I'll first kind of fill you in or discuss more generally examples of infectious epidemics and how warming can work to fuel these. Secondly, I will focus on the work that we're doing now on eelgrass meadows and the continental scale nature of that work. And then, lest we all be too bummed out and unhappy about all these really grim things, also in the context of the COVID outbreak, there are things we can do to improve ocean health, and I want to end talking about that. So this is a paper that was led by Jeff Maynard when he was a postdoc in my lab. And what I like about it is that he reviews most of the major cases where a marine disease is temperature sensitive. So these are cases where these diseases will increase with warming. And of course, prominent among them are things like corals, and also our eelgrasses, as I'll say, and also our sea stars. But there's other cases like the withering syndrome of abalone, oysters, and lobsters. And in fact, the interesting thing that Jeff did in this paper is actually forecast when a lobster disease was going to change from Long Island Sound to affect Maine lobsters. Another one of the projects in this is work that was done by Allison Tracy, who is a PhD student in my lab. And the question that I'm asked almost the most often is, are these marine diseases increasing, or how do we know if they're increasing? Well, that's actually a very hard question because we don't have baselines. And so we entitled this paper, The Elusive Baselines of Marine Disease, because we worked out a method for kind of going back in time to get some estimates about how important these might be. And so what Allison has plotted here, this is the sum of normalized disease reports from 1970 to 2013 for all the major groups of marine taxa that we could get enough data on. And that's a very royal we. It really was Allison, Wren, and Maddie that did all of this huge amount of work. And so you can see, for example, with corals that for that 40-year period, maybe only 2% of the reports, all the reports on corals were about disease. Or for fish over that same period, maybe 5% of all of the literature was about diseases. But if we break this down, what you can see with the coral example, this was the one case where we detected a significant, not only detected a significant increase in the levels of disease, but could also assign some of the causal factors. So here you're seeing the plot from 1970 to 2015 disease reports per taxon. You can see that it's up to 15% as it's increased through time at its peak. And what we have plotted here are the percentage of reefs with temperature stress. And so we're able to actually define that warming events are facilitating some of the outbreaks of coral disease, which was kind of one of our hypotheses. I'm kind of not doing this study justice. I'm just really kind of brushing over it, but I wanted you to understand that we've been taking this sort of broader look and trying to assign causes through time. And at least for something like corals, we can certainly identify a big increase through time that's related with warming. And I'd love to talk more about the corals. That's an entire separate lecture, but I'm just going to leave you with this, the fact that we do have these mysterious coral outbreaks that are expanding. This is one that kicked off about 4 years ago and has been first expanding throughout the Florida Keys and now basically is attacking through the Caribbean. And unfortunately, this is an extremely destructive wide host range disease. So here's a coral. This green part is the living tissue. The white is the part of its skeleton that's actually dead. It's been killed by this very fast acting infectious agent that's at this point is thought to be a bacterium, although they haven't identified the full species yet. In many cases, we don't know where these things come from or when they're beginning. In this case, one of the hypotheses is that ballast water could be one of the culprits here. Now I want to talk about something that I bet most of the people in the audience have had experience with and are interested in, because this struck our shores. In fact, it almost began on our shores. So I want to talk about the seastar outbreak, the wasting disease, seastar wasting disease. One of the things you may not realize is that this is cataloged as the largest marine wildlife epidemic, or epizootic as we call it when it affects an animal. It's the largest because there were over 20 species of stars affected, so a very, what we would call a wide host range, and then a wide geographic range because it was our entire west coast, actually also the east coast, and actually some sites in Asia. And so that's pretty big. And of course it's continuing, and I'll come back to that. It's also important because of the ecological relevance of the taxa. And then there was a warming link with this as well. So that you don't need to listen to me tell this whole story, this is a quite nice video abstract that was produced by the Hakai Institute, Hakai magazine, that tells the story about this paper we published last year entitled "Disease Epidemic and a Marine Heat Wave Are Associated with the Continental Scale Collapse of a Pivotal Predator." And of course, probably many people in the audience know this predator well. This is the sunflower star Pycnopodia helianthoides. And it's one of the 20 species that was affected, but it's the one that's probably been the most impacted. So here we go. Let's listen. Hopefully this comes through.
There's an epidemic in the ocean. Since 2013, a viral disease has been turning sea stars in the Northeast Pacific into melted piles of goo. Of the 20 or so species of sea stars affected by the virus, one of the hardest hit were sunflower stars. Until now, we haven't known just how bad the decline was, but new research has begun to reveal the longer-term continental scale impact of the epidemic on certain species. Scientists in the US are now suggesting we formally list the once common sunflower star as an endangered species. Trained citizen science divers from California to Alaska counted sunflower stars on over 11,000 dives, while scientific divers from the Hakai Institute carried out more detailed surveys on the BC Central Coast. When they looked at all the data, scientists noticed something in common where they saw outbreaks of the virus: anomalously warm water. We still don't know how these warm water anomalies and the virus interact, but researchers say these warmer than normal water temperatures were related to dramatic sea star declines. While divers can patrol waters near the surface, we didn't know whether sunflower sea stars might have found refuge at deeper depths. But thousands of NOAA bottom trawl surveys have revealed that when it comes to sunflower stars, the disease didn't stay in the shallows. For example, data from Washington State shows a crash in populations in both shallow nearshore and deep offshore environments after the epidemic began in 2013. Data from other areas on the coast are similar, with population declines of as much as 80 to 100% in areas across the 3,000 kilometers from Alaska to California. Sea stars may appear to be
Oops, okay, I'm going to stop that now just because we're a little bit time limited, but I hope that gave you a sense of that work that we were able to do and the importance of studying a really difficult species to study like the Pycnopodia, because of course it's subtidal and it requires all diving research to do. So that work was based on both the NOAA deep trawl data, but also citizen scientists. And so if there's any of you folks in the audience that helped with the citizen science collection of those data, thank you, because we could never have accumulated the data on a continental scale to be able to show the impacts. And here's another piece of the citizen science data. This is from Reef Check, California. And what you can see, again, an advantage of these citizen science data is they go back to 2007. And so long before the actual outbreak began. And so you can see here, in this case, there used to be a fair number of sunflower stars. We know there used to be a lot. We would all see them diving. And then this crashed to virtually zero in California, starting in 2014, and very low in our waters. Here's the ecological effects of it. In some locations, sea urchins have exploded. And it's thought that that's because the Pycnopodia were eating them. And when you remove the predator, it released the prey. And they've exploded, causing areas called urchin barrens, because urchins like to eat kelp and algae. And so there are places where the kelp beds have been decimated by these marauding urchins. So it's an example of how we can have an ecological knock-on effect, a sort of a domino effect from the loss of a pivotal predator. So just to give a little bit of an update on what I've been saying, we're now finding that the Pycnopodia are rare or imperiled in many parts of its range. Of course, there's other species we're interested in, such as Pisaster, which is shown here, the ochre star, and Evasterius. All of these species are still experiencing mortality events. This has not gone away. However, we're hopeful for some recovery in the ochre stars. There are some sites where they're doing quite well. Actually, there's some near Bellingham, apparently, where they've been doing very well. Our sites are not replenished. They're still about 30% of where they were, but at least we have stars on our shores. And there's some interesting studies showing evidence that Pisaster at least has a degree of resistance and is kind of tolerating what may be in the water. We've now formed a group with the Seattle Aquarium and the Nature Conservancy California, and we're developing a Pycnopodia recovery plan. I know it seems crazy for those of us that know how abundant the star was, that actual attention is needed to think about whether it needs to be recovered in some way. A component of this, funded by the Nature Conservancy, is actually captive breeding, and Jason Hodin at Friday Harbor Labs has been a master of breeding the sunflower star and producing little babies, which are now a couple centimeters in diameter. So an impressive advance. Sarah Graham and Sarah Hamilton led the listing with IUCN, a red list assessment imperiled based on the fact that it's diminished 97 to 98% in the continental US West Coast. That obviously includes our waters. And if you compare all the way up into Alaska, which they did, it's a 90% global decline. So that's kind of what's going on. Now you're going to have a chance. Yes, all you people that I can't see in the audience, I'd like you to have a chance to think a little bit about what I've been saying. And so I'm going to do a polling. And so I'm going to show you 3 questions. And I think Janine's going to have a polling thing show up on your screen, and then you can vote for which of these you think is the answer. So the question is, why is the sunflower star uniquely imperiled? Okay, of all the species that are affected, it's the sunflower star that we're calling out as potentially in danger of extinction in some locations. Is it that its habitat as a subtidal star puts it more at risk? Or is it that its food puts it more at risk? Or is it that it's the most susceptible to a multi-host pathogen? So it says here, host and panelists, you guys can't vote, but all of the people in the audience, just go ahead. This is not really a hard question. It's kind of just to give us a chance to talk a little bit about it. And for me to know that you're actually there and you have some thoughts about this, because that would help, because all I'm seeing is my screen. I don't see you. So Janine will tell us when we've got a bunch of people that have put their answers in. You're not going to be graded. There's not a wrong answer. It's just kind of interesting to see what you all think. Give you a chance to put your 2 cents worth in.
We've got about 89% folks who voted so far, so almost everyone.
Wow, well, I think we can put the answer up then.
All right, so 74% said it's the most susceptible to a multi-host pathogen.
Wow, you guys got the right answer. Thank you. That's what I think. We don't really know about the answer to the habitat. It's possible there's something in that, but we really do think that it's just uniquely susceptible. The reason that we chose it, and I think maybe in my next slide, I'll talk a little bit about the causative agent. The reason we chose it in 2014 to do experiments on what was causing the mortality is because it was the most susceptible. And as disease ecologists, we know how difficult it is to run inoculation experiments with live pathogens. You'd think it would be easy to make something sick that is susceptible, but it can be hard to figure out how to actually deliver it in a way that your study organism actually contracts the disease. So we worked with Pycnopodia. And from our experiments, and this was published in 2014, there was evidence for a viral sized fraction as causing the lesions. This continues to be very controversial. There's a lot more work that's still needed on this to confirm that it's actually some kind of a virus and that there's still an infectious agent today causing this mortality. Okay, I'm going to move on. But it also is the case that these multi-host pathogens are the most dangerous. So, for example, if you think about, you may not know the story, but if you think about avian birds, well, all birds are avian. If you think about avian malaria and the impact it's had on birds in Hawaii, it's caused several bird species to go extinct because it can live in the more tolerant hosts and just continually be exposing the susceptible ones. Now, of course, we're all dealing with a multi-host pathogen. COVID is a zoonosis. It originated from bats. And as we know, as we've heard, surprisingly, it affects also things like mink, large tigers, and small cats. And so, and as well as there may be other things we don't know about. And we can sort of see obviously why that's a problem, because if you live near a mink farm, you're going to perhaps be more susceptible to picking up a new variant. Okay, enough about that. So I want to move on now and talk about our work in eelgrass meadows. And that's because this is such a vital habitat for us. It's also, as a disease ecology project, it's much more experimentally amenable than the seastar one. So I kind of like to talk about it because I think you can learn a lot more about how this works. And then at the end, I'm going to talk about kind of the strange irony that not only are these eelgrass meadows under threat from a disease, but they also have a capacity to reduce pathogens in the ocean. That's what it looks like. So when a seagrass has this particular pathogen that I'm going to talk about, it creates lesions like this that's dead plant tissue. And then this is the zone of the active infection and the plant fighting back. Here's a little lesion here, here, and here. The pathogen is a protozoan. It's called Labyrinthula zosterae, and I'll tell you about how we figure that out and how abundant it is, unfortunately, in our waters. And then we've done work with artificial intelligence that's allowing us to develop a protocol that can screen over wider latitudes. This is what it looks like. So this is the Labyrinthula zosterae. These are the protozoan cells. These are actually very, very tiny, running around in their mucus network inside. So here they are inside the plant. There's its chloroplasts on its surface. And they're destructive, okay? So it eats up the plant and causes these lesions all over it. And this is Maya Groner. She's one of our colleagues that's led a lot of this work as a postdoc and now as a research scientist. And here's what one of these lesions look like. We've spent well over a decade on the diagnostics so that we can confirm that these lesions are actually caused by that particular agent by culturing it, by getting pictures from histology and now molecular tests. And I'm going to take you through a little of that because I want you to be confident that we know how to identify and diagnose this. So this is what we're faced with. We're faced with a bunch of plants and we have to decide if they're healthy or diseased. This one's pretty easy. It has these very characteristic lesions. This was a little harder. At first, we weren't sure, but it turns out these are early infections that haven't yet caused necrotic tissue. And they're the ones that have some of the highest loads of the cells. Okay, this splotchy stuff, this was confusing at the start. It turns out to just be desiccation. When the plants get overheated or exposed to too much sunlight at low tide, they just get all blotchy. That has nothing to do with this infectious disease. Again, here's more of the infections. And again, more on this one. You can also see how nice these scans are. This is what we use for all of our surveys to keep a record. And here's the data that from the qPCR test that Morgan Eisenlord, a PhD student in my lab, and Colleen Burge, a colleague, have developed. What you can see, here's this little black lesion here. Very high cell count in these solid lesions. So this is over 600 average Labby cells per milligram of leaf tissue. These ones that have started to go necrotic have much lower levels, so the damage has already been done. Same thing here. These look the most dramatic, but there actually are very few infectious cells left in those. And then these gray tissue, there are no cell counts because it's not caused by Labby, and the control tissue is all clean, 0. So just to give you a sense of how we do that. The other way that we confirm that we know how this disease works is through inoculation studies. And I want to take you through a really beautiful experiment that Phoebe Dawkins and Morgan Eisenlord did. And what they did is they inoculated experimental plants and then grew them over 20 days, and then kept an equal number of control plants that were not exposed to the live culture. And so if there were any lesions on these control plants, you'd see the little pink showing up here, and there isn't any. So the control plants stayed clean for the whole 20 days. The experimental plants started to show big lesions after 2 days that continued into 20. Morgan, oh, and that's what these look like. So here's this plant number 202 at day 5, 12, and 20. So you can see this lesion began. This is from the inoculation, a bath inoculation, and then grew through time. Similarly, this one developed multiple spots, which then coalesced and then grew. And then here's the qPCR, the molecular results that Morgan ran, showing at 2 days, there's up to almost 8,000 cells on average in these plants. Again, 0 in the controls, and then up through time. So over time, the little bits and pieces show up inside the controls, and that's just because there's a little bit of an infection that carries over that was brought in from the field, but it's nothing compared to the ones that are inoculated. So that's why we know what we're working with. We can't say that there aren't some other disease agents, and we're interested in that. But I think in terms of these characteristic ones, we have good methods. We've been doing surveys in the San Juans, and that's kind of the center of gravity of our research project, although now we've expanded out because we've found such high levels. And we've been interested in how temperature can affect this. The whole reason I started working on this disease is because it was reported to be temperature sensitive. And so 10 years ago, I was concerned that with warming, we might expect it to get worse in our waters. Here's the 2015, 16, basically the blob high temperatures. You can see the waters are much warmer with a several degree temperature anomaly here in both the winter and the spring. And what we found, and the reason I'm showing you the winter and spring values, is that the prevalence of disease in any one summer, the percentage of eelgrass blades that have lesions is dependent not only on the temperature that summer, but also what happened the previous winter and spring. And so we're finding that there's an important effect of that reservoir as the plants are overwintering. And so we're going to go back and do more work with that. So this just shows over time, this is starting in 2013 at all of our sites. And you can see there's been an increase into 2017. This is the percent of blades. So at some of our sites, it's almost 100%. It's actually shown as the proportion, but it's also 100%. This is the severity. You can see again, we had a big increase into 2017. That's also gone on into 2018 to increase. And then the most concerning is that we found big reductions in the density in our meadows. And so we're trying to connect the dots to investigate whether the disease is actually causing the decline in the eelgrass and some of, well, most of our intertidal sites. So at some of our intertidal sites, we've found 1 or 2 meters of the top of the intertidal is basically gone. So we wanted to study this over a wider range because Zosterae marina occurs on the entire West Coast, and it's a vital habitat on the entire West Coast. And so we really needed to know if it was just problems in our waters or it was more widely distributed. In order to do that and have the proper replication, we survey thousands of plants at all of our sites. And so it's just too much work to all hand score ourselves. So we've trained this image segmentation program to recognize and quantify these visual signs. And I can't tell you how much work actually went into the training. This took years of training to make sure that the program was doing it properly. And it basically, again, I'm doing a terrible job of explaining this because I'm not a computational biologist, but this was developed by the same people that work on how Google self-driving programs recognize cats so that your car stops if it's self-driving. Anyway, in the end, the average error is just as good as humans. And we can now run all of our latitudinal images in a couple of minutes. And so here's the results of that. And I want to acknowledge Lillian Aoki. She's the postdoc that helped run all the data from all of our sites. So this is a large project that is run by Emmett Duffy at the Smithsonian with a whole bunch of partners along the West Coast to collect samples from all of these 6 sites. And I can show you now the disease prevalence at all of those locations. And interestingly, somebody might have accused me of thinking that our site had the highest disease, but I did not score these. Elisa scored these. And it turns out that our location has some of the highest on the West Coast. You can see also high in Alaska. Again, the Alaska and BC sites are virtually pristine. So this was a concerning and unexpected to us level of disease at these sites. We're also monitoring the area of the beds with drones, and I'm not going to talk about that today. So I just wanted to show that. The other thing that this allows us to do is to check on the temperature relatedness. And so Lillian has just finished the analysis to show that the higher levels, again, this is disease prevalence, that's the percentage of individuals infected. It's actually shown here as a proportion, so it should be, again, 100% against the temperature anomaly. A temperature anomaly, for example, in this case, a 0.5 degree temperature anomaly means this site was on average at that 1 pixel spot, 0.5 degree warmer than for the last 20 years. To calculate that. And so what you can see is this is suggesting at these local sites, the warmer locations are having higher levels of disease. So this is only our first year of data in a 3-year study. So we'll have more in the next couple of years. So I'm going to stop talking about kind of the detailed part of the survey work we've been doing on eelgrass diseases at this point. And what I'd like, this is one of our plants that we've been following through time. I'm going to just show you some of the take homes for what I've just said, and then you're going to have a chance to poll. This is actually polling 2. You're going to have a chance to poll on these. So let me just first summarize these points. I talked about our diagnostic test and our ability to culture allow these inoculation experiments for phenology, which means that allows us to tell how these diseases develop through time. Our artificial intelligence machine language learning lesion recognition allows continental scale surveys. We've shown that in the field and lab, and I didn't have time to show you the lab work, and that has been published, that these infections increase with warming. And we have some evidence to suggest that our declining seagrass bed density is related with disease severity. Now in the poll, you're going to have a chance to pick which one is most interesting to you. And it'll kind of be fun over this whole group of you to hear, to see what you think. So if Janine wants to go ahead, and there she goes, it's launched. So just go ahead and vote for one of these. Again, it's obvious there's not a wrong answer here, but it is interesting to me, and I think it will be interesting to all of you to just see what people think about what they've heard. And of course, we can talk about some of this later. We'll have some time for discussion. And I bet you're really fast this time because now you've had experience with how to do these polls, and I bet it won't take you any time at all to get this done. Oh, actually, you know what? I just noticed that this was not intended as a trick, but I've swapped in a different one here. The disease has waterborne transmission. I didn't actually talk about that, but now that it's there, after you're done the poll, I will tell you about our evidence for waterborne transmission, or maybe while you're working on the poll. What we've done is we've actually outplanted sentinel plants different distances from seagrass beds. Because we want to know whether this disease can transmit through the water or whether it just requires contact. And rather surprising to us, the sentinel plants that we put 400 meters, that's 1,200 feet from the eelgrass beds, actually picked up as many new infections as the plants we put in the beds. Okay, so I bet we're about ready. I can't wait to see the answer. So you thought, let's see, I'm not sure what that means. Does that mean, 13, let's see. Oh, okay, got it. 9% thought the qPCR was interesting. 35% were interested in the machine language learning and allowing us to do continental scale. A bunch of you, 43%, were interested in the warming. And then surprisingly, a fewer number said you were interested in the waterborne transmission. That's fair enough, since I didn't tell you about that before the poll. But anyway, that gives us sort of some things to think about. And the qPCR is hugely important to us, but I can understand maybe that's not the most interesting thing. But of course, you need to know that we have a reliable method to do our work. So I want to move on to the third part of my talk. And I'm intending this to be a more positive. This is a solution-oriented part in the book. This is one of the solutions I talk about for disease outbreaks. And this is work that we did in Indonesia as part of over a decade of work that we were doing on the health of coral reefs in Indonesia. And this particular project was led by then postdoc Jolie Lamb, who's now a professor at UC Irvine. And she picked up on some of the work we've been doing on marine hygiene and was able to quantify that pathogenic bacteria can be reduced by 50% in Indonesian seagrass beds. And I'll show you what that actually looks like. And then I'm going to tell you about some work we're doing more locally on this. And so this was the title of that paper. This was published in 2017. So we were working on Bering Lompo Island, And in the book, I tell some stories in the book about, because we've actually done some very exciting work, including a lot of the work we got to do in surveys all over locations in Indonesia, but particularly on this island. But we were running a workshop for Indonesian scientists about how to identify coral disease and species. And then we all ended up getting sick, and quite sick, like with typhoid. And so it kind of raised the question about what was in the water, what was on land. Now you can see from all the houses with no septic that it's kind of obvious why people got sick, but we were also interested in what was in the water. And so our first cut at that, if you look in the pink boxes, this is the number of bacteria of Enterococcus in places where there were no seagrass beds. So all those little red dots are bacterial indicator bacteria. And then where the seagrass beds were present, very few by comparison levels. And so this kind of led us into a really big analysis over 4 different of these islands about how this gradient from shore through the seagrass beds to the reef changed. And what we found consistently for this indicator bacterium is that the levels were very quickly diminished in the seagrass beds compared to outside locations paired right next to it. It's getting ahead of me. And then low down to the reef. So I'm just showing you this indicator bacterium. Jolie and colleagues went on in this science paper to sequence all of the bacteria. And again, the same pattern showed up. There was a big, roughly 50% diminishment of some very pathogenic bacteria in the seagrass beds. Jolie then went on to try to make the link with the environment, to bring together the notion that human health and coral health are intertwined in their environment. And so she surveyed coral disease. This is black band disease and white syndromes show the biggest effect. You can see the marine disease, the disease prevalence outside the seagrass bed is roughly twice or 3 times what it is inside the seagrass bed, and the same thing for this white syndrome. And so we were interested in this sort of hygiene effect of seagrass beds. So it really I've been thinking a lot about this and this idea that you could use nature's superpowers to reduce disease risk, not just for corals and maybe sea stars and things that we care about in our biodiversity, but it's also pretty relevant for humans. And so I wanted to do this same study in Seattle's waters, and I don't have all the results, but this is a study that was actually funded by CDOC to work with the Mussel Watch folks and outplant mussels at a whole bunch of ranges in Seattle and inside and outside seagrass beds. And then leave the mussels there to pick up the bacteria and then sequence them after a period of time. And so I can't show you all of the human pathogens, but there were a lot of very bad things that were present in those mussels, things like Campylobacter, Staphylococcus. And of course, we know that this water is periodically bad. King County's been fined for sewer overflow violations in the rain. And so despite the fact that we have good sewage treatment in our waters, sometimes it just gets out of hand. And so in the numbers, there's a lower diversity and number of the human pathogens in the mussels that were inside the seagrass than were outside the seagrass beds. And so this is not published yet. This is work that's kind of hot off the press and that we're just working to get finished, but it's looking like a pretty good study. So I'm pleased with this. This is still preliminary. This is kind of the way forward to be thinking about natural ecosystems. It's not just seagrasses. Of course, any kind of plant can potentially detoxify pathogens. And maybe in the discussion, I can talk a little bit about what the cause might be. I'm going to skip this just because I can see I'm running low on time. So all of this brought me to writing this book. And I don't know how many of you have tried to write books, but it's not really very easy to think about how to write a book that somebody would want to read. And especially in this case, I was thinking about, oh my gosh, there's so many organisms in the ocean affected by disease outbreaks. These are just a few of them. Which stories do I tell? How do I tell that whole story? In a lot of cases, these disease outbreaks disrupt food chains. We've talked about how species can be driven to endangerment and even extinction. There's human and environmental causes. There's also good things. There's curiosity, there's adventure. There's some really cool scientific discovery and innovation that's wrapped into these detective stories of how to do this work. So I was a little bit overwhelmed, but I was thinking about Michael Pollan's book, Botany of Desire, where he picks 4 plants and tries to tell the whole story of humans' interactions with plants based on 4. And I thought, oh, that's what I'll do. I'll pick 4 of the big outbreaks and I'll use them to tell a larger story. For example, the corals allow me to talk about how disease can be a global threat to marine biodiversity and of course, climate links. Our starfish outbreak with the keystones being toppled in an ecological domino effect. I didn't talk about abalone today. It's actually one of the most exciting stories because it took so long to figure out the causative agent. And now there's a phage that's attacking the bacterium that's attacking the abalone. So, I mean, you know, the plot just always gets thicker and thicker. And then, of course, salmon are very important to us, and salmon viruses have been a big deal. So I included that as one of my 4. So I hope some of you might be interested in reading more about it.
Yeah, we could go on, but running short on time. And I think maybe that's a good segue to say, yeah, you should go buy the book. Absolutely.
Thanks, Stefan. And I just want to end with thanks to the you know, I told this story, but these are the people that did the work. Super, super talented people that I still have the pleasure of working with. So
Yes,
thanks.
yes.
Yeah, I'm done.
Always. Well, yeah, always. We've got time for a couple of quick questions. One of them that came in early and you sort of addressed it, but the question was, how much of the uptick in disease in the last few decades can be accounted for in terms of improved detection methods versus actual increase of disease prevalence?
Yeah, that's a really good, yeah, of course. That's really a great question. And I mean, we can't really know the answer to that. With corals, again, that's a very good example because we know that the corals that are being killed by these infections, we wouldn't have 500-year-old corals if they had been being affected at this level for the last couple hundred years. And so in that case, it's pretty clear cut that these are really, you know, ravaging diseases that are taking down the ecosystem, but certainly some of the increases. And, you know, in that paper that I talked about, not all of the groups showed an increase through time. I mean, that's again, kind of what was interesting about that paper. There are cases, for example, some of the turtle diseases have actually gotten better through time. So it's not a uniform increase. And I'm sorry if I implied that. I did go through it quickly through time. But yeah, good question about that. Thanks.
So the question is, it's interesting that the level of disease in eelgrass is related to sea surface temperature anomaly, not absolute sea surface temperature. Do you think that is because of genetic resistance in the sealgrass at lower latitudes?
Yes, that is, somebody's definitely paying very close attention to what I said. So thanks for that excellent question. We think that's really interesting. And that's one of the reasons to do the continental scale, because obviously the absolute temperature is much warmer in the southern part of the range. But the fact that it's the anomaly is suggesting that these populations are in fact somewhat locally adapted. And when their environment changes where they live, that's what triggers. From that figure, Oregon was the one that really was fascinating to us because the Oregon team kept calling us when they were doing their surveys because they were supposed to be collecting lesions that we could do qPCR on there. Like, we can't find any, you know, what do they look like? We can't find them. And then when you look at their data, that's the place with the cold temperature anomaly and very low levels of disease. So that was really quite interesting. And again, it's the first year, so we're anxious to see how this is going to go through time.
My apologies, there's a whole bunch of questions coming in, but let me end with, what makes you hopeful when you think about the health of the Salish Sea?
Well, what makes me hopeful is the people here, all of you, because I can just tell from those great questions that you know, you understand all of this well. We really are people of the sea here, and there's an extremely high level of interest and attention to ocean issues here. And I think that that is what's going to allow us to be an example for other places. We're in a tough spot, right? We're going to be dealing with climate warming. We're going to be dealing with overpopulation. And so we're going to have to move fast to compensate for those factors that are going to be pummeling our marine ecosystems. But I think with the level of engagement we have, and the love for the Salish Sea and just the value. I mean, we just have the most staggeringly fantastic, you know, marine resources virtually in the world.
Yeah, yeah. Again, my apologies to all the questions that came in, to be continued. Let me put a quick, you said we're people of the sea, which is a great segue for next week's talk. Ruth Miller is speaking on salmon people, tracing indigenous resistance across the Pacific Northwest. So I think that'll be another great talk. Drew, thank you so much. Fascinating. I would love to just keep going. It's great stuff. Thank you very much.
Yeah, you're welcome, Stefan, and thank you, and thanks to all of you for coming along with me. And if you have other questions, I don't know, you can email me or something. Appreciate your interest, and can see it's getting dark here, but yeah.
Okay.
Thank you very much.
All right. Thanks, everybody. We'll see you next week.